Alright, welcome to the pre-lab information for the HPLC lab. We'll be doing the actual experiment part of this lab during week 2 of the lab. So just as an overview of this lab, so first we're going to start by diluting out our fruit juice samples. So they're appropriate for reading in the HPLC. Then we'll make up some vitamin C standards, and then we'll make up some other standards and blanks. We're going to syringe filter all of our standards and samples directly into HPLC vials. We're then going to load those HPLC vials onto an instrument tray and run the HPLC. At the end, you'll get a whole bunch of data from your run that you will then be able to analyze. So just as a general precaution before we begin this lab, something to keep in mind the whole time is to please think carefully before executing each step in the procedure. There's better to ask a question before trying out something than to potentially make an error that would cause you to have to start over, in particular because we have limited extra glassware. So if you have to repeat a preparation procedure, that could potentially be an issue if we don't have enough glassware. So please be very, very careful. As to the third bullet point here, precision here is very critical. Please make sure to use really good laboratory technique, especially with working all of this different glassware and equipment we're going to use during this lab. Another precaution is to please make sure you label everything clearly. You have to be able to keep track of which solutions are what, because at the end we're all just going to be putting them all into a bunch of vials. So make sure you know exactly what's in each of all of your containers and all your vials. And finally, at the end of the lab, please save your solutions. Don't discard them. Just in case we need to check or rerun any of those samples. Alright, that being said, let's talk more about some of the equipment we're going to use in this lab and some of the measurements we're going to make. So first, here's a little introduction to how to use a volumetric flask. So if we're planning to dissolve a solid into a volumetric flask, we actually want to measure that solute that we're going to dissolve directly into the flask. We don't want to weigh it on like a separate piece of weight paper or weighboat and then transfer it in, because we could lose some material on the original material that we weighed it out in. So if we weigh out directly into the flask, we won't lose any of our sample. And then once we've measured our solute into the flask by weight, we'll then fill that flask partway with the solvent. And just to note here, most of the solvents you're going to use for this lab are already prepared for you. They're designed to work especially for use with the HPLC. So at any rate, if you're dissolving a solid, you're going to measure the solid into the flask, fill the flask partway with your solvent, maybe up to about here. And then at this point, you're going to want to cap the flask and mix it really well until the solute is fully dissolved. So this could mean some shaking, some swirling, some inverting, whatever you got to do to really fully dissolve that solute. And then once it's fully dissolved, continue to now add solvent until the bottom of the meniscus reaches this line. So not my best picture of a meniscus, but you get the idea. So with the volumetric flask, in case you haven't used them before, they are specifically designed to very, very accurately measure one particular volume. So all volumetric flasks will have a line somewhere on the neck of the flask that tells you exactly where that volume is inside the flask. And so in particular for HPLC, we're making some very, very extremely precise measurements. And so we want to make sure that all of our weights and volumes are pretty accurate so that we can then trust our results when we get them at the other end of the HPLC. So we're up to step 4. So we added our solvent until the bottom of the meniscus reaches the line on the flask. And at that point, we're going to want to cap again and mix it very well. So this means at least 6 inversions with shaking. Again, you don't want any undissolved bits. You want everything to be thoroughly and uniformly mixed. It's very important to have a uniform solution with very, very minimal amounts of any particulate matter, because we do not want to accidentally introduce any particulates into the HPLC. A note for how to cap the flasks. So the best option you have for capping the flask are these ground glass stoppers, which you see in the picture up here. And make sure you use these ground glass stoppers for your standards as well as your juice. We also, in case we run out of ground glass stoppers, do have some plastic snap tops, and these can be used for some of the other solutions you're making where you don't necessarily require the very best seal. However, just keep in mind that these snap tops can be really tight, so be very, very careful when you're removing them from the flask, because we don't want to break the glass and have anyone get hurt. So that's the volumetric flasks. Another way we're going to be measuring very exact volumes in this lab is by using volumetric pipettes. And these are a similar principle as with the volumetric flasks. Volumetric pipettes also have one line drawn somewhere on the narrow part of them, and they're designed to measure exactly one volume very, very accurately. So just a couple of notes about how to use the volumetric pipettes. You're going to want to rinse the inside of it with your solution, or at least the solvent that your solution is in, before you use it. And this can help you to measure more accurately by sort of helping to make sure that your solution doesn't end up clinging to the sides of the pipette. It'll already be sort of wet with the solvent before you use it. And then once you're ready to actually fill it with your own solution that you're measuring, you can use either the rubber bulb or the pipetters with the thumb wheel. You can just make sure you're using an appropriate volume for the volume you're trying to fill into the pipette. As you fill it with liquid, you'll fill it first to above the mark, and then you're going to want to carefully lower the level of liquid until it reaches that exact line, which I did not draw accurately, but you get the idea. You're going to reach this exact line. And when you're dispensing from where you filled slightly above the mark to the actual mark, that can go into a waste container. And then when you're ready to actually dispense out the exact volume of sample, you can then dispense it fully into your container that it's going into. So now here are some more notes on how to specifically prepare some of the solutions you're going to be using for this lab. So one of the solutions you'll have to prepare is your juice analysis solution that you're actually measuring in the HPLC. So you were asked to choose a clear juice because we don't, again, want any particulates running through the HPLC, but just to make sure there aren't any solid sort of bits in your juice, the first thing you're going to do with that juice is take 5 ml of it and put it into one of our centrifuge tubes and centrifuge it down in one of the benchtop centrifuges that you've used in previous weeks. At that point, you can take the supernatant, just the clear part of the juice. You can discard anything that pellets out. Take that supernatant and start to create a 1 to 10 dilution of it by using a 10 ml volumetric flask. And so you can do that by taking 1 ml of your juice supernatant, put that into the volumetric flask, and then you're going to add the dilution solution that's already prepared for you until the total volume reaches 10 mLs. So that makes a 1 to 10 dilution because you're going from 1 ml of juice to 10 mLs of total volume. And you can label this sample juice. So the other one of the other samples you're going to have to prepare are your vitamin C standards. So first we'll start out by creating a stock standard, which is 1000 ppm of vitamin C. So to do this, you're going to use the volumetric flask, just like we described at the beginning of this video. So first you'll weigh out 50 milligrams of vitamin C directly into your 50 ml volumetric flask. And this way you don't lose any vitamin C. If you were to weigh it in another container and then transfer it over, you could lose some of that vitamin C that you measured. So make sure that you're using a balance that has at least 4 decimal places to get some precision. Also keep in mind that you do not need to have exactly 50 milligrams. An appropriate vitamin C weight can be anywhere from 47 to 53 milligrams. We want you to measure that precise weight down to about 0.1 milligram, but it doesn't have to be exactly 50.0 milligrams. It can be anywhere in this range to give you approximately 1,000 parts per million. And then one other note, make sure that you're reading the balance correctly. Be aware of whether it's set to measure out grams or milligrams. That's very important to be measuring out 50 milligrams and not 50 grams. So once you've measured out that vitamin C, you'll then add some of your solvent into that volumetric flask, dissolve up that vitamin C, and then fill up the total volume to 50 mLs for your 1000 part per million stock standard. Right, that's your stock. Once you've made that most highest concentration stock standard, the next thing you're going to make is an intermediate stock standard. And this one is 100 parts per million vitamin C. So since 100 is 1/10 of 1000, what you're essentially doing is making a 1 to 10 dilution of your stock standard, which is exactly what's happening here. So 100 parts per million out of 1000 parts per million. So again, we're going to take another 50 ml volumetric flask. We're going to take 1/10 of that volume, which is 5 mLs. So you're going to take 5 ml of your stock solution. and then you will fill up the rest of the volume until you get 50 mils total volume. So 5 and 50, that's a 1 to 10 dilution, giving you a concentration of about 100 parts per million. Once you have that intermediate stock standard prepared, you're now going to make your working standards. And these range from 10 to 100 parts per million vitamin C. So I just want to note here, your top working standard, your 100 parts per million, that's the same as your intermediate stock standard. So you don't have to make a special dilution to get your 100. You're already there with your intermediate. But to prepare your other ones, you're going to take a number of different, 10 mil volumetric flasks, and then weigh out either 1, 2, 4, 6, or 8 mils of your intermediate standard. And then fill the rest of the volume up to 10 mils with your solvent, your dilution solvent. And just a reminder, your highest working standard, which is your 100 parts per million, is the same as your intermediate stock. But anyway, once you've prepared these working standards, please label them L1 to L6, where L1 is your lowest working standard, that's your 10 parts per million. L6 is your highest working standard, that's 100 parts per million. And you'll also have 20, 40, 60, and 80 in between there. So all those will be between L1 and L6. Something to note, just for your information, your standard L3, which was 40 parts per million, this is what you're going to use as your identification standard. It doesn't matter for right now for when you're preparing these standards and dilutions. It will matter later in your analysis, so just keep that in mind that your 40 parts per million, which is your L3, is your identification standard. So now let's talk a little bit about preparing your blanks. So there's a few different blanks that are very important. The first one is the column void time working solution, and this is H2O2 in TFA. And this is something you can measure directly into the HPLC vial. It's already prepared for you, and it's something that helps us measure the void time. So the way this works is that H2O2 essentially travels through the column at the same rate as the solvent does. It doesn't interact much with the column. And so if you measure the time it takes for the H2O2 to move through the column, that helps you measure out the time it takes for solvent to travel from one end of the column to the other, which is another word for that is the void time. So how long does it take to empty out the column once if you just had solvent running through? So that's your column void time working solution. Your next blank that you're going to prepare is your element blank, and this is going to be your standard and sample dilution solution. You're going to, again, measure that directly into an HPLC vial. You can call this your blank element, or BLK-EL. And again, I have all of these in quotes. There's ways you can label all of your different blanks, because please make sure to label all of your different vials. There are going to be a lot of vials. So these so the column void time working solution and the element blank, these solutions have been basically prepared already for you, so you can put them directly into HPLC vials. For some of your other blanks, you'll have to do some measurements yourself before you can put them into the vials. So the first one of those is going to be your sugar with element blank. So here we're taking our here we want to measure, so our element blank is just a blank, so we shouldn't see any peaks when we run it through the HPLC. We have our sugar with element blank, and this blank will give us information about what a peak for sugar looks like as it comes off the HPLC. So we do that by taking a sugar TFA solution, again, that's already been prepared for you, and you're going to dilute it 1 to 2. And you do this by taking 5 mils of that sugar solution. You put it in a 10 ml volumetric flask and fill the volume up to 10 mils. So you've gone from 5 mils of a solution, you doubled that volume, total volume up to 10, so it's a 1 to 2 dilution. And you can call this your sugar blank. And then we also have some more controls to prepare. So our next control is our sugar standard that's prepared to match our sample. And so this one is going to be similar to our sugar blank that we just prepared. We're going to take 5 mils of that pre-prepared sugar TFA solution, and we're going to put it into a 10 ml volumetric flask. But to this particular sample control, we're also going to add 4 mils of our intermediate standard. And this is going to give us the equivalent dilution as our 40 ppm working standard, which is our L3. And so you can call this your 40C plus sugar, because 40C meaning 40 parts per million, which is what you get when you have 4 mil of your intermediate standard diluted into 10 mil total volume, and it's got sugar in it. And so this is, so this will help you see what it looks like when you have vitamin C. So, okay, so your blank that we prepared on the last page with sugar will have just sugar. And then in our sugar standard prepared to match our sample, we have some vitamin C in with our sugar. So we should see now a peak for vitamin C. So it should look different than our sugar blank alone. And we can now see which peak now represents vitamin C. And then the last control you're going to prepare is your own fruit juice sample, and you're going to spike that with the standard solution of vitamin C. And so for this, you're going to take 1 ml of your juice sample and 1 ml of your intermediate standard, and you're going to put those into a 10 ml volumetric flask and dilute to 10 mLs. You can call this juice plus spike. And so what this can tell you is you want to make sure that the difference you see when you add a known amount of vitamin C to your juice, you want to make sure that this difference in peak area or measured basically concentration of vitamin C looks the same when it's in a juice solution as when you just have vitamin C kind of by itself. And this helps you find out if there are other components in your fruit juice that might be interacting with the vitamin C and perhaps would alter the measured concentration that you see. So if you have a known amount of extra vitamin C that you're adding, you can then see how that looks on top of your juice sample. All right, so those are the controls you're going to prepare. And so just a reminder, for your sugar standard that's prepared to match your sample with the vitamin C in it, that vitamin C concentration is equivalent to the concentration of your LC working standard. And for your spiked fruit juice that has a little extra vitamin C in it, the equivalent concentration of that extra vitamin C is the level of your L1 working standard. So now let's talk about the next step. Once we've prepared all these solutions, before they go into HPLC vials, most of them will have to get filtered. And to do that, we're going to use syringe filters. So in these pictures, first, this picture on the left shows you just a regular 5 milliliter syringe. There's an area on the bottom of it that's threaded. It's called a lure lock, and it allows you to screw in other components to that syringe, and that way they won't get pushed off the syringe when you push the plunger. So syringe filters are one thing that you can screw into that lure lock area, and that's what we see in this middle part of the diagram. This little orange attachment is attached through that lure lock system. And then on the right here, we have our HPLC vial, where you will be depositing all of your samples. So let's talk a little bit more about exactly how to do syringe filtering. So first, you want to fill up your syringe with your sample. And you want to make sure that there is no extra air. You can do that by filling the syringe and then turning it upside down and tapping it and trying to remove air bubbles. You can also directly pour. You can just pull the plunger off the back of the syringe and just pour your solution in if you feel comfortable doing that. Overall, you'll want, for each of your vials, you'll need about 1 ml total of your solution. So when you're first filling your syringe with sample, maybe fill it up with about 2 ml, and that way you're leaving some extra wiggle room for waste or for loss. So once your syringe has been filled with your sample and the plunger is back on and it's ready to dispense, before you dispense, you want to first put the syringe filter, and you want to attach it to the end of the syringe with a twisting motion. It'll take about a quarter turn of twisting. Then once everything is assembled, you have your sample inside the syringe with the filter attached. Please be careful. Do not point your syringe and your syringe filter at anyone. Syringe filtering is an opportunity for pressure to build up inside the syringe, and we don't want either end of the syringe to accidentally pop off due to a pressure buildup and hurt anyone. So please, once your assembled syringe and filter are ready, hold them vertically. Once you're ready to dispense, the first thing to dispense, you'll just press the syringe plunger gently. This is going to allow the sample to go through the filter and then into the container. If you have enough excess, if you actually put about 2 mLs of sample into your syringe, you actually want to try to discard the first quarter to a half ml of your sample. And this can be just in case there's anything on the syringe filter or in the syringe itself. Discarding the first small amount of sample that will help you discard any any particulates or any contaminants that are on the syringe filter itself. So once you've discarded that first 1/4 to 1/2 ml in a waste container, you can then put the rest of your sample into the HPLC vial. And please be careful as you're depressing that syringe plunger. Be aware of the pressure. If you feel a lot, so there will be some pressure. You have to push to actually, push the liquid through the filter, but you don't want too much pressure. And if you feel that back pressure increases by a lot, just go ahead and switch out the whole filter, because maybe the filter got clogged, maybe there's a lot of excess particulates in that particular sample, or any other reason. So just change out the filter. You don't want to actually press too hard, because this can actually break the actual filter housing. And if you do that, the filter isn't actually filtering anything anymore, and you can potentially add particulates and other contaminants to the HPLC vial, which is the whole what we're trying to avoid by syringe filtering our samples, right? We want only clear liquidy liquids in the HPLC. We don't want any particulates. And so just be aware of that pressure and be very careful with your syringe filtering technique. And so if a syringe leaks, just go ahead and give it to an instructor to be disposed. Once you're done, do not dispose of any of your functioning syringes and filters. We'll have a designated location for you to put them. Do not throw them away. All right, so at this point, you've filled up your HPLC vials with your filtered solutions that you push through the syringe filter. Once your solution is in the vial, you'll want to put the cap on the vial. So you want to tighten the vial cap, but don't over tighten them. They can just be sort of finger tight. You don't need to put any sort of big effort into it. Just make sure it's closed enough. It can potentially cause problems if they're closed sort of too tightly. So at that point, you can place your vials in the instrument tray, and you can do this in the order matching your sample set method, and this will be given to you in the lab. And so in this list, we have a list of all the samples you're going to be loading. This is not the order of the samples you're going to be loading, but this is just make sure you have all of these samples. All right, so at that point, you're sort of the physical part of the experiment is sort of done at this point. You load every of the vials onto the tray. The tray is loaded into the HPLC, and the HPLC runs all the samples. At that point, after the HPLC is done running, you're going to get output in a number of different formats. So the first type of output you're going to be getting is chromatograms. Chromatograms are graphs of absorbance at one wavelength versus time. So these will look a little bit like this, where you have absorbance versus time. And these chromatograms are going to help you to monitor the amount of material that's exiting the column at different retention times. So what does that mean? So at one particular absorbance, if the material coming off the column absorbs at that wavelength, you will see a peak when that material exits the column. So you might see a low amount of absorbance, and then an absorbent material comes off the column, you see a peak. Maybe a few minutes later, you see a second peak. Maybe you see another peak, who knows, etc. And the amount of time it takes to see that peak is the retention time. And each material that goes through the column in those particular conditions is going to, each particular substance that goes through the column in that set of conditions is going to have its own unique retention time. So the retention time you measure tells you what material is being measured by that peak. So in this first peak, it took this long to come off the column. That's time 1. This one is time 2. That's the retention time. And then the absorbance level that you read, and in particular the area under the curve, this is more indicative of the quantity of material that came off the column. So a bigger peak with more area under it means more stuff came off the column, and how long it took for it to come off the column tells you what that stuff might be. So that's one type of output. Another type of output that you'll get are spectra. And these spectra are graphs of absorbance versus wavelength. And so unlike with our chromatograms where you're measuring at one particular wavelength, where you know that your sample that you're interested in measuring, you would want to measure at that wavelength where you know it's absorbing. Spectra, on the other hand, will look at all the wavelengths and tell you where there are high absorbance levels. And so these types of spectra are good at also identifying different materials that might be coming off your column. And so if you see an absorbance spectrum, usually they're a little bit high at the beginning, and then depending on what material you have, you'll have a peak, and the location and the shape of that peak can tell you what material it is. And so if you measure, for example, at each of the peaks you saw in your chromatogram, each of these peaks indicates a different material. You can then look at the spectrum you created for that one peak based on time, and now you can get a full wavelength spectrum of whatever material was being measured by that peak. And if you look more carefully at that, that can give you a better idea of what the material's identity is, because most materials will have a unique UV absorbance spectrum. And so, yeah, and another way that these spectra can be used is to pick which wavelength you want to use for the analysis. So if you just had, let's say, we're looking at vitamin Z, you might just want to first obtain a UV-Vis spectrum for vitamin C to see which wavelengths produce the strongest absorbance, and then you can pick this wavelength, whatever wavelength number that is, to be the wavelength that you measure for this chromatogram, which measures over time at one wavelength. So that being said, our last type of output is the standard curve. So this is going to get you a graph that tells you the peak area versus concentration. And you're going to specifically use this for your vitamin C working standards. So here you have peak area. Here you have concentration of vitamin C. And so, so you're going to have, depending on the concentration of vitamin C in each of your working standards, the more vitamin C you have, the larger the peak area. And so for each of your 5 or 6 standards, I forget how many, you're essentially going to get a number of coordinates on this plot where the higher the vitamin C, the bigger the peak area. And you can essentially draw a line of best fit through those standards and find the equation for a line. And then in this case, the Y is your peak area, and the X is your concentration of vitamin C. And so you can use the known information about both concentration and peak area to create your equation for a line. And then you can use the peak area information that you gather from your juice sample, which has an unknown amount of vitamin C, and plug it in as the Y in this equation to calculate how much vitamin C was in your juice sample, as well as in your other solutions. So with that being said, now you are ready to tackle this HPLC lab. Good luck.